speaker join us today. And for that, I'm going to ask Dr. Todd Gould to give an introduction.
I'm super excited to introduce Dr. Fernando Goes, who comes to us from across town at Hopkins. I've known Fernando's work for quite a while, and it's really exciting that in just about the past year, I've had the opportunity to collaborate with him on a couple of projects. So at Hopkins, he's an associate professor. He's also director of the Mood Disorder Clinic. He's co-director of the research program of the Johns Hopkins Mood Disorder Center. And relevant to this presentation today, he's the director of the Precision Medicine Center of Excellence in Mood Disorders at Hopkins. So in terms of background, he's been at Hopkins for a while. Actually, you got your undergrad from Brown, and then you did your MD at Hopkins, residency in psychiatry at Hopkins, mood disorder fellowship at Hopkins, and rose now through the ranks in faculty and now director of a couple of clinical centers. His main clinical interests and focus is on biological underpinnings of severe mood disorders. This includes bipolar disorder and major depressive disorder. He's authored over 100 research articles related to these interests, particularly also subtypes of mood disorders, especially early on, was very interested and involved in the psychotic subtype of bipolar disorder and genetics that underlie that subtype. He does work in the phenotyping of patient populations and also has done quite a bit of genetic work at Hopkins. He's a PI on a number of grants, including an NIMH study of the genomics of the anterior cingulate cortex in major depressive disorder. He's also a site PI on a couple of treatment studies with ketamine and one that compares ketamine to ECT. So Fernando, thanks for joining us this afternoon.
Okay, can people hear me on the Zoom? Great. Thank you, Todd. Oops, I just lost the screen. I just lost both screens. Oh, it's back. Great. Yes, I've been at Hopkins for a long time, but I do have divergent opinions from the usual. So we're neighbors, of course, and hopefully we can become closer neighbors. I've always wondered why University of Maryland and Hopkins don't work a little bit closer, and I think I'm very excited to be here and to be working with Todd and hopefully to start more of this collaboration. Okay, precision medicine in mood disorders. I have no disclosures. Okay, briefly, I'm going to introduce the concept of precision medicine. I'll talk a little bit about genetic studies because I sort of think of genetics as the background. It might not be the primary driver of precision medicine for some of our disorders, but I think it's something you always want to know, and it's a good example of how to study something comprehensively. And in genetics, when we think about precision medicine, it's really about polygenic risk scores, and I'll describe a little bit what I mean by that, and discuss some potential near-term implications where we might actually see them clinically, and talk a little bit about this precision medicine center that we just started at Hopkins. This will be mostly aspirational. I'll talk about some of the sort of driving initiatives and studies that we're doing. It's mostly clinically translationally focused. So yes, I wanted everybody to keep calm. Yes, there's a paradigm shift going on, but it's it's going to be inclusive. Hopefully we have to be very deliberate, and those of us who are clinicians have to remember that we need this paradigm shift to be clinically useful, and that's challenging, and that has a lot of opportunities. Okay, so you all know this. I won't spend much time, but the burden of mental disorders is tremendous, particularly mood disorders, early onset. The problem is it starts early and it's recurrent or often chronic. Usually in our mood disorders clinic, we see a lot of chronicity that's multifactorial, but still is associated with a lot of disability, both in males and females. And beyond this sort of chronicity, there's also the burden of increased mortality. Now, some of this is due to suicides, but most of this is actually due to mortality due to medical causes and just sort of poor lifestyle, poor living conditions, and things such as cardiovascular disorders. But there is this sort of steady rise of suicide rates, and this is a few years old now, but the CDC actually just came out, I think, a month ago with this year's rates of suicides, and the suicides continue to have this very slow rise. So we don't think that there's anything related necessarily to the pandemic, but it is this sort of conglomeration of many sort of sources of distress, including mood disorders, that's really leading to this increase that still hasn't budged, despite a lot of attention, right? A lot of attention. So where I come from, one of my interests is, so what's the problem here? The problem is just like in all of medicine, and I do want to couch this in all of medicine, because we're tied. It's not that psychiatry is any different. There is this sort of problem of our treatments being imprecise. That is, not everybody responds to them. And in fact, in terms of antidepressants here on the right side, we see that the average effect size is modest as a whole, right? So what this cries for is better treatments and also better subtyping. And better subtyping is essentially the ethos of precision medicine, right? So what does precision medicine? Everybody has their own interpretation. It's more or less the same. Here's from the FDA. And you can see in that final sentence, the goal is to target the right treatments to the right patients at the right time. So a more tailoring type of approach, thinking about individual vulnerabilities as well as risk factors, right? And in my title, I say, is this any different than any type of medicine? I mean, before the word precision came along, obviously it's been in the dictionary for a long time, but before it's become a buzzword, would we think of it any differently? And no, we wouldn't really think about any differently. It came along in 2015 because of this gentleman, or this ex-president. State of the Union Address 2015, so 7 years ago. Tonight, I'm launching the new Precision Initiative to bring us closer to curing diseases like cancer and diabetes. I wish he had also put mental depression, for example, in there. Give us access to personalized information that we need to keep ourselves and our families healthier. So a focus of precision on common disorders, as well as more of a preventive focus. And shortly after the State of the Union address, Francis Collin and Harold Varmus in the New England Journal gave us fleshed out a little bit more about what the NIMH thought about precision medicine and what it would entail. So it's more preventive. It's more sort of thinking about prevention, although treatment strategies are still front and center. This word of individual variability, right? So what does that mean? So individual variability, because we have now new data from all these sorts of forms of assays and methods to characterize biomarkers, right? So these large-scale biological databases, genetics is part of this, but it's not just genetics. It's all the omics, as well as the computational tools that you need to try and put this together, to analyze it and try and put this together. So it's this confluence of all these sorts of things that makes it, that leads to the why now question. And I put this last quote because people think, of course, precision medicine is primarily going to be DNA, genetics, and RNA. But even from the very beginning, I've highlighted behavioral data is there, right? I think we're going to have a very limited view of precision medicine if we think of it just in terms of DNA and genomics. So behavioral data, and we in psychiatry, right, should be leading that aspect. I mean, typically, it's complex data, and at least in our department, we haven't specialized too much in this notion of behavioral phenotype at the level of humans, not just animals. But this is data that's going to be particularly important. So with the Precision Initiative came this All of Us initiative, right, which is a volunteer-based biobanking that we're trying to mimic on a small scale at Hopkins. Anybody can join. It's pretty easy. I've done it. You spit in a tube, send it off, and they send you some more information. You agree to give your medical records, which I had a little bit of pause in the beginning, but ultimately, I think medical records don't tell for most of us is not something that I worry necessarily about sharing. Again, it's an individual decision, so if you're comfortable with it, it's pretty easy to do. And since I'm from Hopkins, these are my two deans, right? That I have to stay on the good side to keep my job. They wrote a brief description of what they wanted out of sort of a Hopkins initiative for precision medicine. And this is where our mood disorder center comes in. We certainly weren't the first kid on the block. I mean, we were the first non-Alzheimer's psychiatry kid on the block, let's say. But of course, many medical disorders are there. And I think part of the importance is, it's very important when there's initiatives for medicine for us to be part of the table. So, but where they address it, so Anthony Rosen and Scott Ziega. Rosen's a rheumatologist, has his own lab. Scott Ziega is a famous biostatistician. So the why now question, right? Raising, because is this always been medicine? So the why now question is similar to what Varmus and Collins said. We have the capacity now for improved measurements. We have these incredible computers, most of them in the cloud, and we have now ways of sort of linking things through medical records, for example, and through the cloud as well. And we'll come back a little bit to talk about the linking aspect of it, which is this sort of learning healthcare systems, which I think actually Marilyn probably knows a lot about. So is this new or old wine in new bottles? And you can ask, haven't we always had subtypes? Have they not always been important for clinical formulation? They've certainly been important for the boards, right? I mean, these things come up again and again. And it's interesting, these are concepts that come up a lot in the textbooks. So things like melancholic versus atypical depression. And of course, when we think of prototypes, you do sometimes see patients, but the problem with these prototypes is they haven't been empirically validated particularly well. And when empirical validation tries to sort of find these clusters, it usually falls a little bit flat. There's too much sharing across these clusters, and we don't have the data to show that they respond any differently to any sort of medications, right? So yes, this has always been a conception of what a clinician should do and what we want to do in order to find subtypes where we have better prognostic and better sort of therapeutic outcomes. But and we've had them, and they're in the boards, right? And so you have to answer as if they exist, but the issue is, there's not a lot of empirical backing. So what do we have to do? To some extent, we have to sort of start from the beginning. So this is a little bit of information showing that it's on uncertain ground. Now, so people are now, so bipolar 2, for example, my ex-chairman did a lot of work in his career, sort of talking about the salience of bipolar 2 in terms of genetics. And 20 years later, has that sort of firmed up the nosological state of bipolar 2, and probably not. We still have sort of thoughtful papers such as this talking about the existential crisis of bipolar 2. And you can make this argument essentially about any of the non-major, I think, psychiatric syndromes. How many of major psychiatric syndromes, it's a discussion, but any sort of departure, there's sort of issues, right? So there's a lot more to know. And on the right side, it's a recent, or not so recent, but 7 years. This was a trial of depression, multi-country trial. That try to find whether the subtypes make a difference in terms of treatment response. And at least as they measured it, these are always complicated questions. Are they measuring it correctly to begin with? But as they measured it, no, there wasn't any difference. If you're initially called melancholic versus atypical versus anxious, everybody tends to improve more or less on treatment with the usual sort of variability. So how do we find better subtypes and how do we find more effective interventions? And this is from, this is an old slide from David Lewis. And I think many of you essentially will know this. The simple answer is you need to understand the underlying pathophysiology and biology a little bit better, right? Or the causal processes of these illnesses. Some of these could be social, some of these could be sort of within the psychological level of analysis, but ultimately it's about trying to understand more of the causal processes. And it sounds straightforward. It's fiendishly difficult because the brain is so complex. Nevertheless, there are times when serendipity strikes, but still we we have to be trained to actually see the serendipity and make use of it. And that's really how we've come up with many of our significant advances. And I've listed some of the major advances that are clearly can be life-saving for patients. On my end, one of the reasons I initially got interested in genetic was this high heritability, right? So this is nature giving us a clue about where to look. Heritability is a statistical construct. I think for if those of you that have worked a little bit in research, you know the limitations of heritability as a term to make certain inferences. But if you haven't, it's a statistical construct, and it's about the two variances. So it's actually a bit of a complicated construct, and it's probably in this very nice review paper from Peter Visham. There's some common misconceptions that sort of help you a little bit with heritability. So high heritability does not imply genetic determinism, right? Just because something is highly heritable in a certain environment doesn't mean that you can't change the environment to change the phenotype of the prognosis. High, heritability is perilous when you try to compare group differences, right? We hear a lot about that, and that's because the groups are under different environments. And low heritability, of course, doesn't also mean that genetics aren't important. So for example, the example is everybody has a neck. So the heritability, so there's nobody that's alive that doesn't have a neck, right? Doesn't mean that genetics aren't important for you for the creation of a neck. And then the issue that at least in, when I started and in sort of my career has really become very evident is that these disorders of high heritability, like schizophrenia, like bipolar disorder, there was initially this sort of hope that even if there wasn't going to be a massive Mendelian gene, that there might be a handful, one or two genes of big effect. But so far we haven't found them. And most of us probably think that it's just a conglomeration of minor minor associations that I'll talk to you about. So large heritability doesn't really mean that you'll find a gene of large effect. You can have just many genes that conglomerate together and have a large effect. And it increases the complexity of the disorder, increases the complexity of the search for the genes and the underlying causes for the disorder, but it doesn't change the heritability notion. So what is inherited? And apologies if this is very basic, but it's mostly DNA. I put the next things there because sure, some of these things are also inherited, that is passed through generations. And once in a while, you'll get a very provocative Nature paper or something like that saying how you've got this epigenetic transmission of this or that and this little tiny RNA that goes through it. But generally, that's exotica, I think. We really need to sort of focus on the DNA. The other things are probably just too unusual to make much of a effect on our phenotypes. Now, in certain animal or in certain plant models, for example, there's interesting biology. To my view of the literature in humans, there's just there's no convincing data that any of these other things are associated with inheritance, right? I know there's some provocative papers out there, but they rarely get replicated robustly, I guess. So genes are helpful because they have stable structure, right? That's what makes them heritable. That's what makes them the ideal sort of heritable unit. We can now, they're finite, they're identifiable. So this isn't this sort of sense of unknown. So in genomics and transcriptomics work, for example, now there's a lot of isomers, for example. And so there's a lot of changes in the expression. Genes, you more or less know what you're going to get. They're now very easily measured in large samples. And then the other thing is what makes them easy to collect and to test is that you can get it in the blood and you can infer that more or less that's the genetics that you're going to get in the brain. Okay. There are a few exceptions, for example, with somatic mutations, but we don't know how important they are for things such as mood disorders. It's looking a little interesting in things such as dementia or in some unusual Mendelian disorders, but otherwise there is this facility that you can get something from the blood. You can get it from spit. You can get it from any aspects of the body you'd like. But the genes are markers, and we have to remember when we think about genes, you shouldn't think of it as an endpoint, right? This isn't. I'll talk a little bit more about what we measure when we talk about genes, but they're markers, and we're ultimately interested in the downstream and the worker bees, which are the proteins. I won't talk at all about proteins because it's very complex to measure them. You need a different specialty, but ultimately think of genes as markers, right? As a baseline for precision medicine, but as markers. So let me go through a little bit of before I talk about in order to introduce GWAS to those of you who are mostly clinicians. So if you get your genome sequenced, and the big actually innovation now is that genome sequencing the price has gone down. It's actually only $200 if you can get the special Illumina deal. When we first did some of the initial sequencing, we did just a handful of samples. It was $10,000. So and that's just in 5-10 years. That's a lot of money that we wasted. We should have waited. So 3.1 billion base pairs in your genome. 1% or so is protein coding. That 1% is worth remembering because that's the protein coding genes that are most relevant, most easily interpretable. But when we do a comparison, right? When always we do a case control comparison, we're looking at variation. If there isn't a variation, we can't compare anything. So what we're ultimately interested is, even though you've got billions of base pairs, we just really need to look at just quote unquote a million or so of the variation. So it's 3.5 million variations in your genome, but there's some redundancy. So ultimately, if we just look around a million, we've got most of the information for these common variants. And I won't talk about this class called structural variants, but just to know that it's actually very important for things such as autism, intellectual disability. It's less important. The reason I won't talk about them is, unfortunately for me, not for the patients, it's just not very prominent in mood disorders, right? So they just don't come up. But in intellectual disability and in autism, they do come up, and they're often associated with cognitive deficits. So we look at the variation. We embrace the variation in genetics, and just a reminder that generally variation is a good thing, right? If there wasn't variation and if we didn't have these little errors, we would still be, I guess, anaerobic bacteria, as Lewis Thomas said, and there wouldn't be any music. Well, there wouldn't be a lot of other things. Most of the reasons that we have variation has actually nothing to do with diseases. This is one of the challenges of studying this. Most of the reasons has to do with your migration patterns of your ancestors. So in cosmopolitan societies such as our own, there's substantial caveats to analyzing data because of this sort of variation. You want to have it as controlled as possible. That can be challenging, but there is a fair amount of analytical tools that can sort of help us figure out the control. So, and we look at variation, but we also have to remember that 99.9% of our DNA is the same, right? Across peoples, across continents, and all this sort of things. Remember also 99% of our DNA is the same with apes as well. So you just need a little bit of variation in certain spots. So let's talk a little bit about genome-wide association studies. It's extreme. I think you see them a lot now, including in clinical journals. They seem pretty complicated, but just remember that it's a very simple comparison at each locus. So that is the position that in the genome, you can tell whether you have an A or a T, for example. Or a T or another T. You're simply comparing the frequencies in cases versus controls. Essentially, that's it. The challenge there is making sure that you're not being misled by a potential confounder. But ultimately, it's a simple 2x2 table where you compare a frequency between cases and a frequency between controls, and you get a p-value. Once you get a p-value, the way that we sort of show is through this Manhattan plot that I'll show you a little bit. And again, it looks complicated, but it's extremely simple, right? It's one chromosome next to another. You start with chromosome 1, You go to chromosome 2, you've got markers all along these chromosomes, and you've got the strength of, you've got a strength of the association. And there is a magical threshold that you need to be above. And that magical threshold is the threshold that allows you to control for all the multiplicity, all the multiple testing. That's this magical 5 × 10⁻⁸. So you can do a GWAS, but if you don't reach that threshold, you're in uncertain territory, because the chances are that you might not replicate this finding. And, we spent many years actually doing GWASs that didn't get to this. Now the GWAS sample sizes are enormous, tremendous. So it's actually not hard to get to 10⁻⁸. The problem is that because the sample sizes are so big. The challenge now is controlling for confounders and thinking that you have an association of the phenotype as opposed to something else that comes along with the phenotype. So as I said, in the beginning, long time ago, it was really hard to get genome-wide significance in the beginning because the sample sizes just weren't big enough. So consortium came up. Now all we do is consortium work. But in 2011, it was pretty novel. And if you start, people needed to get along, groups needed to get together across countries, across disciplines, and across departments. And if you put things together, you start to get genome-wide significance. And this is, I'm showing you the example for bipolar disorder. It is absolutely the same for any other disorder of medical phenotype. You collaborate a few more years, you get more genome-wide significance, you keep on going, you increase the cases and the controls, you get more genome-wide significance. And this is the latest in bipolar, for example, 42,000 cases, you get 60 or so genome-wide significance. And again, work that's in progress now, which has been boosted by this 23andMe company that some of you might know, and some of you actually might have given your samples to. Now the samples are enormous. 23andMe has millions of controls that you can add. 23andMe also has people who declare that they've been diagnosed with bipolar disorder. So this is 64 genome-wide significant cases, but the latest sort of PGC GWASs are well into the hundreds now because of this enormous influx of 23andMe. It brings up this interesting question of how comparable the two, and I won't talk much more about it, but just to sort of mention that it's not fully comparable, right? So it is a question worth characterizing. So the challenge, of course, is that the odds ratios, and this is just what nature has given us. The odds ratios for these associations are pretty small. That's why you need 42,000 cases. That's why you need 300,000 controls, right? And that's what makes further downstream biological interpretation challenging. And that's what makes clinical application or translation of GWASs also challenging. Okay, so for example, TRANQ-1, this is just the top association in bipolar disorder. It's got an odds ratio of 1.1. So that means it's found in 51% of cases and 48% of controls. So we all have these risk factors. When we think about the risk factors, these are very common, and they're shared across cases and controls, shared across ancestries general, with some exception. Okay, it's called the Manhattan plot. Because of the World Cup and because I'm from Brazil, I like calling it a São Paulo plot. So Manhattan, you used to see all these buildings, right? Now all these GWASs are filled with associations, and it looks like this concrete jungle, which is my hometown, for good or bad. So GWAS works essentially for everything, truly for everything. What you do with that is another question, but you increase the sample size, and if it's got any form of heritability, you get genome-wide association. We struggled with getting barely anything. We increased the sample size. We've got a lot. And then now we sort of want to give it to people like Todd, but they don't like it because it's too much. It's just that, they want 1 or 2 or 3 genes. We're giving them hundreds. And so there's a lot of challenges and unhappy people with all the numbers of GWASs. So, what can you measure? So this, terrible thing, male pattern baldness is pretty heritable. Miserableness, right, in the UK Biobank, just by asking people, do you tend to be miserable? You get lots of genome-wide significance. Just by asking people, what kind of cereal do you get a genome-wide significance. This is a little silly, but you get to the point that now the sample sizes are so big that you have to think about epi. Like, what is different about people that eat cereal, cornflakes versus not, right? So clearly it's not about the cornflakes, but probably something SCS, or could it be something about, lactose, or could it be something about carbohydrates, or could it just be, something related to where cornflakes is available versus not? There's all these sorts of things. So tongue in cheek here, but when you've got a really strong biological signal such as rheumatoid arthritis, it overwhelms, right? So it's not. I'm being tongue in cheek in a sense because even we're getting a lot of genome-wide significances. Now it's difficult to interpret, but really when you've got a strong signal, it comes up. I think I've talked a little bit about some of the sort of issues where I think the main win from GWAS is this, what I call this initial inference about potential pathophysiology, right? Not as clear as we would like it. And sometimes very challenging to go from a certain locus to a gene, as I'll talk about in a little bit. But it's this initial empirical inference that you can be pretty sure about from human samples, right? Then you've got to do a lot more work. The work certainly doesn't stop. The other challenge is that both in humans and in cell models and in animals, these associations tend to be pleiotropic, right? They're associated with diverse phenotypic associations. So I think when I was there was always this sort of question about, how do you validate a psychiatric phenotype? And I think when I was a student and a resident, there was this desire that I think genetics would ultimately help us validate. But if these associations are shared across phenotypes, not completely, but often it's more the rule than the exception, that's going to be challenging for us to use them as a validator. But again, that's what nature has given us. And that's also why I started by saying, think of these as markers, right? The phenotype is very distant from where you start off in terms of the genetics. So there's many ways to modify it, right? One association to get to a brain-related phenotype, there's so much potential ways of changing, of affecting, of expressing things in this way or another. And can then be used clinically. And I think I gave you the example. This is another one, and it's a very clear no. No, GWAS can't be used clinically, not at the single level. The effect size is just too small. Okay, so that took a few years to kind of get over this complex about finding small effects, but I'm over it now, and there's still love that you can get from small effects. If you're more of a mindfulness, so Mother Teresa also liked the small effects, right? So it's adding small effects. So small effects also gives us strength. But here's a scientific reason why small effects can still be pretty heuristically useful, is that they can identify drug targets, and we have examples. That is, now these are drug targets that we had drugs prior to the GWAS, right? So they're nice examples, but there are two examples in cardiovascular disorder that identifies when we look at GWAS of heart attacks, we find drug targets to some of our most effective treatments, and so we're hoping that in our GWASs in psychiatry, we'll also get that level of insight. And then GWAS is important for drug discovery. So if you have an association to a phenotype, chances are that you'll have an approved drugs are higher. So odds ratio is usually around 2 here. So it doesn't give you complete confidence, but I think drug companies increasingly want to have genetic association data before they embark on a very complicated drug discovery or drug development program. But I said that it's a challenge. So you've got a GWAS. I'm going to talk about many challenges, but that's because there's lots of opportunities, and we need lots of smart people to join and work in this field. So it's challenging to go from a GWAS from an association to a gene because there's lots of correlation in the genome, and because the genome actually is fiendishly complicated as well. It's not just one straight line that follows simple rules. It convolutes, it turns around. It's got lots of proteins. Sometimes where you are in the nucleus makes a difference. Sometimes how you're twisted makes a difference. And all of these have potential regulatory ability, and of course, the environment and other proteins can affect that. Just because we're thinking about genetics doesn't mean that other aspects of the cell don't affect it. So the more we know about genes and gene expression, the more potential sort of interesting ways of regulating genes we discovered. And what this means though is you get a locus, for example, when I show you a locus, when you see a locus, usually there's a gene next to it, but really think of that written in pencil and very likely to be erased, because you've got this, for example, locus here. It could be any of these potential genes. It could even be a gene, pretty far away. So suffice it to say that it takes a lot of work to go from locus to a specific gene, and that's active work. And for example, that's work that we're doing, and sometimes, and this is a work that really where you get some resolution by looking at this process called triangulation, right? You've got one association, you want to see consistency across different modalities of studies. So for example, this is the GWAS for one association, and we have a colocalization here, for example, for an EQTL. That is, it's also this locus, and these markers are associated with higher expression of this gene. So this is nice. Again, it doesn't nail it, but this is sort of gets you along the thinking more about the causal path. Okay. So this is a slide really just for, as I said, was for Todd and for his lab, just for him to understand that we also do some kind of complicated genomic stuff. There is convergence. I'll talk very briefly about this. There's just, so where do we find some of this triangulation? Partly is we want to see what kind of converging pattern do we see with gene expression data. It's challenging, but what I'm showing you here, and just very briefly, is that there's converging to genes that are expressed in neurons, and in particularly in glutamatergic and inhibitory neurons, right? So the GWAS and the gene expression data set that we're working with both show some element of conversion to these important classes of cells. These are very, admittedly, these are very important. They're probably much of the brain, right? So it doesn't necessarily narrow things down, but you're thinking about all this new drug discovery that's coming up in depression, right? It's glutamatergic NMDA receptors and inhibitory, so GABA receptors. Think about the new drugs that are GABAergic as well. So this is my most technical slide. Excuse me for just this one slide. I'll go back. Okay. So returning to the GWAS, because I do want to talk about where can we think about GWAS in terms of precision medicine? And I was saying that at the single level association, you're not going to get anything that you can hang your hat at because of the effect sizes. But if you add up, think about the Mother Teresa quote. If you add up all of these genome-wide significant associations, and if you add up, you can even go down lower, these subthresholds association, you can then get a score that's called a polygenic risk score. And that's what might potentially eventually trickle down to the clinic. So just to give you a little bit of an example. So you've got all these loci, these associations, right? I showed you the Manhattan plot. Again, it's slightly more common in cases versus control. You can just add them up. It's really as simple as that. Of course, there's control for potential confounders, and you can weigh them in different ways. And sometimes you can give them more of a weight if they're in more functional regions of the genome. But ultimately, the concept is just about adding it up. And if you add it up in cases and controls, what you usually find is that they're slightly more common in cases versus controls. That's your polygenic risk score. Usually in the question then with polygenic risk score, do you just look at the genome-wide significance, so 64, or do you look at the top 10,000? Do you look at the top 100,000, right? There's varieties of thresholds, but the thought about what a polygenic risk score is just simply an addition of risk factors, right? Each of the molecular risk factors. Sorry, this is a MAC to PC issue. So you were going to see very nice curves. Maybe here. They disappeared. Sorry. You were going to see very nice curves that look like this, right? So for example, if you've got bipolar disorder and you actually look at the polygenic risk score, you'll find that in bipolar disorder, the normal is projected low. Okay. So it doesn't make, so if you're here, you've got a score that cases and controls typically tend to have equally as frequent, right? So the issue with polygenic risk scores is that it's probably going to be most relevant for people at the extremes. If you've got an extreme score, chances can be pretty high that you'll have something like bipolar disorder, but that score is going to be pretty rare in the population, right? So for the unusual person, This might be an important informative information for most of us, and for most patients. We'll be living here in the center. And so apologies, it was two beautiful curves. And I gave this example. One of the issues is that the curves are overlapping, right? So again, small effect sizes with polygenicity, we can have larger effect sizes, but still there's a fair amount of overlapping. This is actually an example from 20, 30 years ago, and this is an example that's widely used. So the reason that I gave this example is just to give you this appreciation that we use this. So in medicine, that we often have tests that overlap. Just because there's overlapping and there isn't clear differentiation, it doesn't mean that we haven't found them to be clinically useful. And this example is actually, I don't know if you know, but if you look at these numbers, it's cholesterol, right? This is cholesterol, people who have had and who have not had a heart attack. And you can see the people who have had a heart attack tend to have higher cholesterol. But that, there's a fair amount of overlap. And there's many people who have not had heart attacks that have high cholesterol. And there's many people who've had heart attacks that have low cholesterol. So this is a complex multifactorial risk factor that we use widely in medicine, that we have drugs for, and that shows this sense of overlap, right? So think of this as an example. So heart disease, without heart disease. And then again, this was sort of, it was going to be a bunch of beautiful curves that showed you separation. As GWASs get more and more informative and larger, the separation of these two bell curves get more and more. So the polygenic risk score currently isn't sufficient, right? But in the next 5 years, it might. I don't know for bipolar, it might for schizophrenia, it might for some other sorts of things. I think it certainly will for certain things like cardiovascular disease. Or potentially for breast cancer. And just to go back to this issue of old wine and right, the polygenic risk scores and polygenicity as a sort of theoretical construct has been around for a long time in our field. And in this wonderful and extraordinary paper from Gottesman in 1967. It pretty much has everything that genetics has taught us, right? This was before any kind of genotyping and any sort of GWAS. So it was an incredible conceptualization of polygenicity and what's important in the field. And I think again, if you want to learn about genetics and risk factors, I think this paper is as good as any. But here's the example of where it might be useful and where it's actually being tested. So for example, in breast cancer. So if you've got high polygenic risk score, super high, right? So above 90% within the population. So pretty high, but 10% is still a fair subset of population. So it's not just one or two people. This is your risk of developing breast cancer over the life course. And all of these studies are from Europe, because of the registry and their ability to follow people. So there's a fair difference, right, between high and low. Now, most people will be here, but if you're at the extremes, you probably want to know whether there's some certain level of actionability here. Right. The question is how actionable is this kind of information, and that's what sort of randomized trials are trying to test now, and will need to test, because we can't assume that just having high risk is an actionable insight for patients. Right. There's many reasons why patients might not want to know this risk, and there's many reasons why patients might not react well to this sort of risk. So clinical trials are very important. And then closer a little bit to our field is Alzheimer's disease, where there's stronger polygenicity than in, for example, bipolar disorder. Again, if you're high in terms of polygenic risk score, this is your risk of developing Alzheimer's disease. If you're low, this is your risk. There's fair difference here. There's quite a few years, right? Without treatment, as we find ourselves in Alzheimer's disease, would you want to know? So this information, for example, is of interest for researchers. It's of interest now in drug trials, right? Because this is potentially a way of stratifying. You're trying to understand underlying biology, more or less. But at the level of clinical stratification, is this something that you might want to tell your patient or not? And of course, we've got this example of ApoE4. This is an unusual common variant that has a strong effect size, and we've had it for a long time. And remember, so here's an odds ratio of 8, which is which is a whopper for a common variant. It's very rare to have an odds ratio that high. It's not something that we use in clinical care. I mean, I don't know if it's changed, but it certainly wasn't when I was a med student or a resident, right? Because there's nothing that we can do about it. So think about risk stratification, but also what are we going to do? Here's an example of where I think polygenic risk scores are really going to sort of fit in for things such as bipolar disorder, which is what they do a little bit is they'll be used in the context of other typical clinical risk factors, right? So they give you an extra nudge, additional independent information from each of these risk factors. These are clinical risk factors that are associated with developing bipolar disorder. So in somebody that comes in, for example, with depression or with a mixed picture, and it's challenging to come up with a diagnosis, you start thinking about some of these clinical features, right? You don't think about it very comprehensively, but maybe we should. And what else does a polygenic risk score do? And you can see that by themselves, they don't do a lot, but they always give you a little bit more of a nudge, a little bit more information than the clinical risk factors, right? So they should be seen as part of risk stratification, part of the comprehensive evaluation. By themselves, they're probably not going to excite that many people unless you're at the extremes, but they should be sort of part of the picture. Okay, so polygenic risk scores also allow us to compare across diagnoses and then give us some sort of nosological insights, which are relatively interesting. The main point is that there's a lot of sharing across many of these disorders and many of these diagnoses. Again, you know, from the clinical side of things, this maybe isn't a big surprise, given how you know some people can start with one diagnosis, go to another, and we find so much comorbidity. But what it tells us is again what we know is that our disorders, as Steve Hyman says, they're grounded in biology, but they're not natural kinds, right? So our nosological tools haven't been able to really cleave our disorders or separate them from others. Where I sort of, you know, also get some insights is maybe that's just not the nature of our disorders, right? I mean, that's just a little bit of a you know, challenge that's going to be too much, right? So this is sort of more thinking in terms of, to some extent, dimensional models of psychopathology. Okay. So we've talked about common variations. I'm going to actually skip some of these slides, just looking at the time. So, of course, there's genome-wide significance when we think about how far do we go in GWAS, right? Well, if you're a sort of maximalist, you really want to say, I want to explain all the heritability. GWAS significance so far doesn't give you that much, but we're able to sort of do some modeling and say that if we get an infinitely large GWAS, we can get, let's say, 30% of the heritability. So there's all of this missing heritability, and where is that coming from? And these are some of the potentials. And one of the places that we've studied is the rare variants. So rare variants are rare because they're not shared across the population, and they're relatively recent in your family line and evolutionary. Why do we study them if they're so rare? Because they have a larger potential to have a large effect size. So they haven't gone through natural selection. So if something has a deleterious and large effect, chances are natural selection is not going to let them become common, right? Your chances are you will have less children if you've got something that's particularly problematic, and that the variant will die with you or will die with a few family members. And so that's how it stays rare. If it just stays in a few people, if there's negative selection. We've only recently been able to, I guess, relatively recently. You know, when I was in college, I did a few of these. These were sequencing gels, and they were really painful, and they had lots of radiation, and I absolutely hated it because of all the radiation and all that kind of stuff. And it was really slow. And then when I was started as a fellowship, we would do this kind of assay, which was also a little bit painful. Easier, because we could send it off, but it was painful because you'd have to, you know, be stuck in a dark room and look through this over and over again. So now we've got we've got this way to do whole genome sequencing. It's actually less than a week now. So this incredible way of using optical technology and new assays. And so we can finally test the Tolstoy hypothesis, right? Which is the disorders that we see, are they mostly driven by common variations, or is there something unique about each individual or each family or each sort of subtype, right? So this is if you open up Anna Karenina, the very beginning, there's this famous quote: all happy families resemble one another. Each unhappy family is unhappy in its own way, right? So there's a reason. So when we see a patient, each patient has its own different way of finding having a disorder. We mostly look at the exome aspects of rare variants because of their greater interpretability. This is a rapidly changing field, but at least as of 5 years ago, it was very challenging to interpret the non-coding genome. It still is. It won't be. This is something that will change in the next 5 years. But as of right now, and again, looking at rare variants was a surprise. And initially, the first surprise was, boy, even normal controls have lots of bad looking variations, right? We all do. There's bad looking mutations in all our genomes, and most of us are lucky that they don't have phenotypic consequences. Like you need to have a pretty bad luck for a variation to actually hit something that's important for a certain disease related phenotype. But otherwise, it's not that just patients have these variants. It's we all have them. Patients might have slightly more, but generally it's not that they have slightly more. It's just that they've had the bad luck of it hitting a certain territory that's important for the mutation. And they're most important in intellectual disability and in autism. But nevertheless, we've taken a stab to look at them in bipolar disorder. And we did this, though, we tried to select for early onset bipolar disorder. And so one of the things and looking for the absolutely the rarest of the variation, which is the de novo, right? So when you have your children, you've got the two parents, each parent passes on their DNA, but there's also a little slip up in the DNA replication machinery somewhere along the sperm to sort of fertilization pathway, where every time you have a kid, the kid also has something like around 60 to 70 new de novos, right? New mutations. And that's every birth. Now, what's interesting is that the paternal age of birth is associated with increase, right? This is mostly because of the age of the sperm. So if you're super old, super old, I mean like right here, you're going to double the number of de novo variants that the child inherits. And mostly, as I said, most people get away with it. I mean, right, millions of births every year, and you need really bad luck for it to hit something that's relevant. But we do find a slight enrichment in a relatively small sample, but we do other small samples have also found a slight enrichment in synaptic pathways here with the rare variants. But rare variant story, and this is my last sort of data genetic slide. The rare variant story has been similar to the common variant study, meaning that you still need large samples and large controls. So we initially were hoping that, in relatively small numbers of cases, and that if we pick the phenotype just right, meaning a classic bipolar, early onset bipolar, very severe, we would sort of enrich for the rare variants. Unfortunately, it's again a numbers game with rare variants. And what I'm showing you is, so far in bipolar disorder, the most interesting association with rare variants is this gene called ACAP11. And it was a gene that really only became significant with bipolar disorder if you combined it with schizophrenia, which is, it's fair, but it tells you that it's not really going to be a bipolar specific gene, right? So pleomorphic issues come up there as well. But it is an interesting gene. And part of the interest is because it's a gene that has protein-protein interactions with GSK3 beta. And of course, GSK3 beta has been thought to be the target of lithium, right? So it is interesting. But so, for example, this was, again, consortiums, many Zoom calls, many phone calls. Our group provided, in the end, 1,000 cases, and in the 1,000 cases, we only saw this variant once, right? So in 14,000, you see it 16 times. So it's 1 in 1,000. that you see a variant that's relevant here. So most clinicians with bipolar disorder might not ever see anything like this. Again, this is why you sort of need large sample sizes. So concluding with genetics before I just end up with maybe 5 to 10 slides, a little bit more clinically focused. Yeah, I sort of see it as a mature field. The next 5 years is going to be intense gene discovery. We've already seen it, but I think within the next 5 years, it will be sort of kind of towards the end of this gene discovery, huge, because sample sizes are getting enormous. Collections are happening now all over the world. As long as there's some sort of phenotypic consistency in these collections, will be, I think, mostly done for most of the sort of elements of genetics. Now, there might be certain variants that you need new technology to pick up. The main goal is biological insights rather than, very sort of deterministic thoughts about precision medicine. I see it as a baseline for personalized medicine. I think it's essential. For some people, if you're at the extremes in terms of polygenic risk score, or if you're extremes in terms of having a rare variant, and for example, in dementia, sometimes we see that, it will be very important for you to have this information. For most of us, it won't, because we'll be middle of the ground, right? And that's a broader principle about many things in terms of precision medicines. And interestingly, it's reverse, it's yin and yang. So when I started, there was a lot of attention on phenotypes and assays were very expensive. Now assays are very cheap. And so the GWAS and the sequences are pretty cheap to do. The challenge now, though, is how do you get these very large scale samples? How do you phenotype large scale samples and actually have consistency with the clinical phenotypes and the domains that we're interested in? So for precision medicine, we're very interested in prognosis, for example, and in drug response. And that's a challenging phenotype to measure. I mean, drug response, that's why we do randomized controlled trials, right? Just because somebody gets better and they're on a drug doesn't mean that they necessarily are responders to that drug. The challenge, of course, in our clinical, in our clinics is people are also on 4 drugs. There are many drugs. So this is sort of the next kind of frontier in terms of the challenges. How do you get these phenotypes that are that will be more relevant and necessary for clinical medicine? That's what I call the new bottlenecks. So phenotypes of clinical and prognostic relevance. I'm going to skip the pharmacogenetics if anybody has an interest, because usually people ask that. And the issue with pharmacogenetics is it's simplistically, it's not prime time. The assays that are available are not good enough. Here's one quick example of one patient that got 2 companies and they said different things, and this is well known, right? So the variability in the assays is still a little bit unacceptable. So the science is not concrete. So for example, this one was same patient called an ultra metabolizer for 2D6. Where it's called a normal metabolizer. So which one do I trust? Actually, I've emailed the company to try and get a. I've asked for the raw data. They haven't sent it to me yet, but I want to get to the bottom of this. So there's just too much of this variability, and the clinical data, unfortunately, just keeps coming up with negative trials, right? I mean, it's not that many trials, but there's 3 or 4. These larger trials just have not shown much of a subtle association. So clinicians always ask about pharmacogenetics. I barely ever use it. There's a few very difficult patients that I might sort of think about, but otherwise I almost never use it. And the funny thing is, because they do so much advertising, we have people in our system that use a lot of the pharmacogenetics, and they like it, but that's because they like the psychotherapeutic angle of it. And we have to be careful with precision medicine, right? The language, patients also like it if they think it's telling them information. But I think the information logy, it has not been that robust. So this is a sort of problem with hyper precision medicine, and this is a little bit of an ethical quandary, I would say, in our field. Okay, so ending less technically and thinking about the precision medicine initiative. I think around a year ago in the New York Times, there was Benedict Carey. Often writes about mental illness, and he had a editorial. This was actually when Tom Insel was writing his book. This is when he published his book, right? The issue, you've got all this cool science, all these cool figures, but how much progress have we made to improving mental health issues now? And of course, we haven't made a lot of progress. So, so for example, in bipolar disorders, it's still a challenging, I mean, in the time, I mean, you know, in the last 30, 40 years, have we made a lot of progress with bipolar disorder? I don't know if we have, right? There's, if anything, you know, there's certain things that have gotten more difficult and certain things have gotten better. So misdiagnosis in early stages, it's just very challenging. People hear about 10 years to make a diagnosis. Sometimes that's sloppy, you know, clinical decision-making. Often it's because the prodromal signs of the illness are just not distinctive. There's just nothing we can do. You all know about this better than I do because you know, Maryland has made so many important advances here, but this issue of comprehensive care for somebody with mental illness, it's such a fractionated system. Even within Hopkins, even within my system, we sort of have silos. Insufficient focus on functional outcomes, of course, right? What do we mean by drug response? Is it just a cessation of certain prototypical symptoms? And there's relatively few evidence-based interventions available. So this is both a research and implementation gap. So in bipolar, for example, of course, we have lithium, which is the gold standard. That's easily available. The question is, do we use it enough? We probably don't. But then there's also psychotherapies that have been shown to be effective, but nobody's trained in a bipolar focused psychotherapy. And unless you've got a psychotherapy researcher in your medical system, or something like that, there's just no training for that. And, you know, it's an issue of incentives, right? And suboptimal adherence to treatment. So can obviously precision medicine works when you have one or two causes and you can target those causes. The question is, can it work for our complex diseases? And I think it can. I think the issue is that we need to have appropriate expectations, right? And we have to, starting out, have appropriate sense of the complexity. Larger sample sizes are going to be needed and more clinical follow-up. We have not characterized our patients and our disorders as well as we need to, particularly in this big data scale and big data world of things, right? We have very limited, somewhat superficial information about our patients, and that's not going to help with precision medicine, right? So that EMR gives us access to information, but we need to augment that with phenotypes that are more relevant. So again, this notion, I think most of you know this because actually, you know, Lisa Dixon is a pioneer in many aspects of Maryland, a sort of pioneer about this. So this notion of a learning healthcare system where we're constantly iterating between information that's relevant for patients, that's relevant to researchers, and that is made available to both the researcher and made available to the clinician and the patient in a way that's digestible and in real time. So we can get this information. It just takes us a lot of time to actually make sense of it, right? We need to get to the point that the information is available in real time. So that's much of what our precision medicine. So finally ending on the precision medicine, we're trying to do a learning healthcare system. We have apps, we've got ways of putting information for clinician support. Eventually we want to build that into treatment guidelines. And we want to incorporate all the sort of elements of these sorts of things. Not for everybody, right? This is not going to be relevant for everybody. But if you have some sort of phenotypes where these things might be potentially relevant. We've started across town with a measurement-based program in mostly in Bayview. This is our Bayview Community Psychiatry. So pretty big. We have lots of patients there. And we've been now going for a couple of years trying to get patient-reported outcomes, for example, as for longitudinal data. Unfortunately, with COVID, the waiting rooms continue to look like this, right? So we've moved from in-person essentially to now trying to reach patients almost exclusively digitally through Epic, through MyChart, and through emails. And it works for some patients, it doesn't for others. So we have to think about better engagement because we no longer have the full waiting rooms. And I think we've come to the realization that, well, we might never have the full waiting rooms again as we did before, right? So the kiosks are not going to work. So that's sort of gotten us more into reaching out, and this sort of information can give us, brief looks at. So, for example, we're interested in bipolar diagnoses and the instability of bipolar diagnosis. And what this is a very simple way of looking. If you come into our healthcare system at Hopkins, And the last time we have your information, which is on the right side, does your diagnosis continue? And what it shows is for around half the people come diagnosed with bipolar and they stay bipolar. The other half have some other diagnosis or lose their diagnosis of bipolar, right? In terms of thinking about clinical improvement and improving the lives of these folks and what they should be treated with, reasons for this transitions are very important. So that's something that we're very interested in. This is something that we see more prominently in bipolar 2, more of an unstable diagnosis, again, leading to this sort of questioning of the nosological, the firmness of the bipolar 2 diagnosis. And overall, but this will give us the baseline for what we're calling this mood disorder precision medicine of excellence. And with this baseline, we can then, have some driving projects or when we think about different grants or different ideas, it all begins on the baseline so that eventually the information feeds in and is there for the patient and is there for the next generation. So we are trying to do polygenic risk scores. So we are collecting blood or saliva. We're very interested in digital phenotyping. So this is the Oura Ring and the Mind Lamp app that we can give to patients when they're willing to use it. We've collaborated with sleep psychologists to try and understand whether circadian functioning and for certain patients that have sleep difficulties, whether actually measuring with a home EEG device is going to be helpful. And of course, this is from one of Todd's paper. We're very interested, for example, in ketamine. What we find similar to other sorts of treatments, around 50% of people improve with ketamine. It's an awful lot of work to get somebody treated with ketamine. Are there biomarkers or potential ways to understand why certain people don't improve versus others? Again, you can make that point with all treatments, but since ketamine is something that's relatively short, relatively easy to determine whether response or not, you don't have to wait months. And of course, Todd is across the street. We're trying to get a resident who actually was at Maryland and now is a fellow at Hopkins, Gustavo Medeiros, to write a K grant to look at EEG, in addition to all these other risk factors, as a means of getting response to ketamine and getting that information earlier. I mean, of course, you get it at the end of 2 months of your treatment, but after 2 weeks, for example, is there a certain pattern that will tell us if you're likely to respond or not, right? So similar sorts of goals, central goals to precision medicine. Okay, let me end, but I'll let President Obama and the grand rounds for me. And the reason is he actually just spoke last month. So I started the presentation with Obama 2015 State of the Union. He's now speaking for private companies, but that's okay, I guess. But he just spoke in this Illumina Genomics Forum, and he was asked about, so what do you think the progress has been in your precision medicine initiative, right? And he says it's held back by creaky healthcare system. So he's excited about the innovation and the new sort of biological insights. But he acknowledges progress has been slower than I'd hoped. Because of this big creaky system that accounts for 6 of the U.S. economy. We have to, wow. So this issue is partly, of course, is about maximizing the data that's collected and is everywhere, right? It's, of interest, credit card companies have data about us, right? Personal data about us. So for example, in the precision medicine initiative in Hopkins, somebody said that in a year or two that he thinks that we might be able to get people's financial data. And initially, I opened my eyes, I couldn't believe it. But the issue is the credit card data is, and every time you do a credit score, you've got your financial data and it's out there in the public. It's being used. But in any case, so the data for the healthcare system siloed, hard to figure out, yes. And this issue of incentives, right? It's a legacy system. All these interests, and there's a huge amount of money at stake. So it's very challenging to make changes and to conceptualize changes. But let me leave you with his last advice, right? Which he was talking about the presidency and his decision making in the presidency. And it's how I sort of think about precision medicine, right? Don't let the best be the enemy of the good. The question is, will this make things better for people? If the answer is yes, let's do it, because better is really good. And better is usually the best you can do. So let's take better, right? When he was talking about his decision making, many assets, many facets, it's complicated. So you don't want to let best be the enemy of the good. Thinking about complex diseases, many aspects, many facets, exactly. Let's just make sure that we have a little bit of better, make sure that it's robust, and just sort of add up all the things that can potentially improve the care. So with that, I just want to acknowledge many people in some of the work that I've sort of shown you, include the postmortem brain is a collaboration with Lieber. Otherwise, these are all sort of my colleagues and friends now, particularly Peter Zandi, who I've worked with for many years, and Jimmy Potash, who's our chair, but was my chairman, went to Iowa, went lots of other places, eventually is back and really is very interested in this concept. So thank you for your attention. Sorry, I went a little long, but hopefully there's still a little time for questions. So thank you.
So we do have about 15 minutes for questions. I wonder, can we get the participants up so again, just raise your hand in the chat or in the Zoom, and then we'll call on you.
Thank you. Yeah, thank you so much for an excellent talk. I see in this polygenic risk score that you're maybe identifying those individuals that are classic bipolar. And those individuals, whether they respond to treatment or not, are easily identified. But I wonder how you can see polygenic risk scores helping us to care for patients who are less classic, and where those patients who are not identified, how this still might apply to them.
Yeah, that's a good question. So polygenic risk score will reflect what your initial training set is, right? So if your initial training set is classic bipolar, which it tends to be, because that's what it's going to pick up classic bipolar. If your initial training set is going to be the more mixed stuff, it's going to pick up the more mixed stuff. So ultimately, the question that we want to answer, though, is not so much, is this person have classic bipolar, but what is this person likely to respond to, right? So we need to then go back to the training polygenic risk score and go back to things such as what kind of signatures help us to know if you're going to respond to lithium, if you're going to respond to something else. So right now it doesn't very much. So polygenic risk score is going to be high in people with classic bipolar, but that's because it's reflective of how we constructed it. So you got to think about a little bit about like this. So in a sense, you need to start with a very broad characterization of bipolar disorder. So then you're able to make these more fine-grained strata, right? But ultimately, think back to this issue of classic bipolar. We really want to link it to new phenotypes that are more relevant for clinical care than something like that. So, but then you have to start with the phenotypes to begin with, and that's challenging. So, for example, we've been part of this consortium for lithium genetics. It doesn't seem challenging, but it's actually challenging to get good lithium data on most people. People have been on many medications. People usually don't wait long enough. You know, what do you define as a good lithium responder, for example, is a question. Is it somebody that's going to respond to lithium in 2 months? Right? How long are you willing to let your patient stay on lithium before you determine that they're not a good responder? And the reality is the response is probabilistic, right? So do you need to wait a couple of years on somebody on lithium? And are we in a system, will patients agree to stay on lithium for a couple of years before you determine that? And well, the healthcare system, certainly the inpatient unit doesn't, right? You have to change your medicines pretty quickly. But yeah, so I think it's going back to this issue that you have to think about clinically relevant phenotypes, and you have to get the genetics of those to begin with. Yeah.
Thanks for that was really incredible. I feel like I left genetics research that's about 15 years ago. So you caught me up. But what I was struck by, and you mentioned at the end, the marginal gains, right? And wondering how we move the needle more significantly. And one thing that occurred to me, talking about the amount of heritability that's explained by a couple 100,000 more.
Yeah.
Also, it still doesn't explain the majority of your earlier slide. And concluded that we should focus our efforts on DNA. We're wrong, right? What if we have marginal gains because we're barking up the wrong tree? You're barking up an entirely different tree.
Yeah, I mean, I guess to go back, I don't think we should only focus our efforts on DNA. But so what if we're wrong? I don't think we're wrong. We have these good examples, right? So I think if they work for other aspects of medical disorders, they should work for us. So I guess probabilistically, I think we've got a good shot. Yeah, the other question is, what if these other things are even more complex, right? What if there isn't a way to get a very sort of clear incremental advance? I think or a big advance. I think we're making small advances and they all make sense. And there's a conglomeration between what we theoretically expect and what we're finding empirically. So I think that's helpful. This issue of, do we need another huge sample size? We don't. Nobody, particularly nobody, I think, thinks we do. There is a lot of collection for ancestrally and populations, right? So non-white populations. That's very important, and that will be helpful for fine mapping. But I think just to go out and collect and just to get genetics, the challenge is these studies tend to be expensive and research is also siloed. I think we miss a lot of opportunities by going out and just quickly phenotyping and getting genetics, right? Getting to the patient is really challenging, and we should get a sort of more comprehensive phenotype from the patient and understanding from the patient, including, for example, psychosocial issues, right? So part of this issue of seeing genetics and seeing the limits of genetics has reinforced, some of these earlier conceptions that we have complex disorders that are very environmentally influenced and sometimes environmentally caused, right? But we don't have clear ways of measuring the environment, for example. That's even harder. So actually, to go back, Todd, to your issues, partly the genetics should have been, is the easier aspect of it, right? But I do think we've got 5 more years of this sort of intense genetic discovery. And then I think after that, it's going to be moved on to the next sorts of things. Yeah. 5 years always becomes 5 years at the end of 5 years, but given all of that.
I had a question for you about, you were talking about the diagnostic stability and you showed the graph. So I think about the EMR where you have all these problems that get listed and it may change. How do you sort that? How do you sort the garbage out of that, or the signal from the noise when the EMR allows so many things to just be clicked that you really can't validate?
Yeah, we've been having this question for a while. So usually the clinicians say exactly that, right? And when it's because we're responsible for kind of the pollution. And then the clinical bioinformaticians say, well, the sample sizes you can always get around. Yeah, I think you can't. I think the issue is EMR will only get you so far, and you have to do this augmentation of the EMR. So, for example, send people either do brief evaluations or brief questionnaires, or send, or ideally the issue is also get as much passive information as you possibly can, because people actually don't like, it's a hassle to answer questions. And this notion that we're going to send patients lots of questionnaires and they're going to fill it out is also not very realistic, right? We start and they'll do a couple and then it gets really annoying. Just think about you and your phone, right? When you do a couple and then it gets really annoying. So what can we obtain as passively as possible is one question, right? And that's the digital side of things. And people are very interested in that aspect. But you're right. I think everybody now has a sense of the messiness of the EMR. It's particularly an issue for our disorders because in cardiovascular disorder, you've got a cholesterol level, you've got the blood pressure, these things are relatively unpolluted. For us, what we're working on now is natural language processing to get information from the notes, but we know that we're only going to get so much. But I'm hopeful that with this information, we can then go back to this issue of a learning healthcare system, and we can improve the primary data that we put in the EMR. Because we spend so much of our time as clinicians on the EMR. And why are we doing that if so much of the information is poor? So hopefully this leads to some improvement in what we put in. So I'm hoping there's some automation of the information that can go in the note. So for example, some things so that we don't have to spend our time putting this information in the notes, and that it leaves more time for us to actually just be in the clinical encounter and for putting more of this information that's relevant, right? So one thing is, we're putting just a few. I mean, the drop-down menus can be terrible in EMR. I'm not being naive about that, but we want to have a few of these sort of tables and drop-down menus. For example, when residents do their initial evaluation notes, they're wonderful notes. They can be very long. In this day and age, it's a lot of copy and paste, but but we want a few things that we want to extract. So we understand that we can only extract a few things, but we want that to be improved. And that has to be through an iterative bilateral process. So this also has to be of utility for the clinician. Otherwise, they're not going to be engaged. Yeah.
I think that's part of the problem. There isn't an incentive to clean up the EMR, because if you're going to be the one that's going to spend 3 hours doing that, then clinically you can't see any other patient.
Yeah, no, absolutely. No, it's true. It's about incentives and who's checking out, right? And it's quality control. Because when I make a billing error, I don't know if I get an email. Like the one time I've gotten an email, like the next day from my hospital administrator was a few billing errors. I'll get an email the next day, right? So there's a tremendous incentive for you to get the billing right. But we want to increase the incentives. And so we have to make your life as a clinician easier if this is going to work. And I'm hoping that there's some automation of information that can go into the note that can then free you up to put in just a few things or fewer things, rather than this very long, we at Hopkins also have a history of doing really long notes. And they're very helpful, but what if we could save a little bit of your time with the notes, so that you can, be trained. It's a whole, you can understand it. It's a system-wide change that needs to do. But boy, there's something off with us and EMR, right? This is a system that was supposed to simplify our lives and was supposed to be better for patients. And it is in certain ways, but my gosh, there's other ways that it really isn't. So there is incentive to change. The question is, how do we do it?
We have a couple minutes left. Did we have any last questions from the audience? The virtual audience? If so, raise your virtual hand. If not, we will wrap up.
Great, thank you.
All right, thank you very much, and we greatly appreciate it. Thank you.